Today I am going to explain about topic of biomolecules. So cell. So cell is a structural and functional unit of organism. In our Earth planet, diversity of organisms are present. All these organisms are structurally composed by the cells. So cell is a structural unit of a organism. All organisms body organized by the number of cells. So 1 to trillions of cells are present. So for example, in human beings body approximately 3.7 trillions of cells are present. So organisms body structurally composed by the cells. So cell is a structural unit of a organism. For living being of organisms continuously requires the energy for carrying out different life activities. So these energy obtained by the performing different chemical reactions. So these are nothing but functions of a living organism. So for living being of organism, what are chemical reactions? Functions requires this all are will carry by the cell. So that's why cell is a functional unit of a organism. So cell is a structural and functional unit of a organism. So within these cell different types of chemicals are present. So approximately in cell more than 5,000 types of chemicals are present. So within cell in organisms body more than 5,000 types of chemicals are present. The science what is deals with about these chemicals, then physical and chemical reactions. Physico-chemical reactions among these chemicals and the entire living process. This is known as a biochemistry. Biochemistry. So biochemistry is nothing but science. It deals with study about various chemicals are present in living organisms body, nothing but within cell. So what type of physical and chemical reaction? Physico-chemical reactions takes place among these chemicals. So these chemical reactions responsible for organism being living condition. But nothing but entire about the living process. This science is known as a biochemistry. Then what type of chemicals are present? So carbon containing chemicals, organic and without carbon inorganic chemicals. Then molecules are present in organism's body. These are mainly participate in metabolism of a organism. This entire chemicals is known as a biological molecules or biomolecules. Biological molecules are biomolecules. So all carbon containing chemicals and compounds within organism's body participate in chemical reactions, nothing but metabolism. This all chemicals is known as a biological molecules or biomolecules. Then all chemicals are present in organisms cell, then ions, molecules, compounds, etc. This is known as a cellular pool. Cellular pool. So cellular pool is nothing but so within cell present what type of all chemicals, ions, compounds, molecules, etc. Maybe living and non-living materials. This is known as a cellular pool. So in cellular pool, two phases are present. So first one is a aqueous phase. Then second one is a non aqueous phase. So in cellular pool, two phases are present. So first one aqueous phase, second one is a non-aqueous phase. So first one coming to aqueous phase. So in aqueous phase, two types of solutions are present. So first one is a true solution. Then second one is a colloidal solution. In cellular pool, so two types of phases are present: aqueous phase, non-aqueous phase. In aqueous phase, two types of solutions are present. So first one is a true solution, second one is a colloidal solution. In true solution, all are homogeneous substances are present. So these substances approximately 1 nanometer are less than 1 nanometer is present. So 1 nanometer or less than 1 nanometer substances are present. These all substances are homogeneous substances dispersed or dissolved in a water medium. Water medium. That means these all are dispersed or dissolved in a water medium. We know well water acts as an excellent medium. So these homogeneous substances dissolved or dispersed in a water medium. This type of substances is known as a crystalloids. This type of substances is known as a crystalloids. This is known as a true solution. In true solution, all are homogeneous substances. These are 1 nanometer diameter or size or within 1 nanometer is present. This type of substances is known as a crystalloids. Next one, colloidal solution. In colloidal solution, non-homogeneous non-homogeneous substances are present. So size of these substances is 1 to 100 nanometers or maybe greater than present. So 1 to 100 nanometers. These are the non-homogeneous condition. So this type of solution is known as a colloidal solution. This type of substances is known as a colloids. This type of substances is known as a colloids. So in cellular pool, aqueous, non-aqueous. In aqueous, true solution and colloidal solution is present. Next one, non-aqueous phase. So in non-aqueous phase, different types of number of chemicals are present. So these chemicals are present as a chromatin, then cell membrane, then cell wall, cell membrane and cell wall. So in non-aqueous, so various different types of chemicals are present. These are present in the form of chromatin material, nothing but genetic material, cell membrane, plasma membrane and cell wall. To know about what type of chemicals are present in a cellular pool or a cell, so we have to perform the chemical analysis. We have to perform the chemical analysis. By performing the chemical analysis, we can reveal all type of chemicals what are present in a cell. So for this chemical analysis, we have to take both the living material and non-living material. So by performing the living material and non-living material in chemical analysis, we can reveal, we can know the all type of chemicals what are present in a cell or a cellular pool. So chemical analysis by take on a living material, then non-living material. So living material may be living tissues, so animal tissue, then plant tissue, microbial paste or veggie Tubular materials. So we can perform chemical analysis on this any living material by taking a small amount of this animal plant or microbial waste or vegetable material. We can perform the chemical analysis. Then non-living material is nothing but earth crust. Non-living material is nothing but earth crust. By performing chemical analysis on both living and non-living material, we can get the same type of the elements are present. We can get the list of same type of elements in both the living and non-living materials. So by performing the chemical analysis on living material and non-living material, so we can get the list of elements, same type of elements in both the living material and non-living materials. But the major difference is the amount of the relative abundance of carbon and hydrogen respect to other elements greater in living material compared to non-living material. So by performing chemical analysis or elemental analysis on living material or non-living material, we can get the same type of elements in both the material. But the major difference is the relative amount of carbon and hydrogen with respect to other elements, so greater in living matter compared to non-living matter. So average abundance of some elements in both the living matter and non-living matter. So earth crust. So non-living matter, then living matter. So non-living matter and living matter. So hydrogen. So percentage. So hydrogen in earth crust approximately 0.1% is present. Same hydrogen in living matter approximately 0.5% is present. So next one, carbon. So in carbon, non-living matter, nothing but in earth approximately 0.03%, in living matter approximately 18.5%. So third one, oxygen. Oxygen approximately 46.6% in non-living, in living approximately 65% is present. Next one, nitrogen. Nitrogen very little condition, very little in atmosphere, not atmosphere, in earth crust. In atmosphere 78% is present. In earth crust usable form, that means modified form, nitrate is very less in condition, so very little. So in living thing approximately 3.3% is present. Next one, so sulfur 0.03%, here 0.3% is. Next one, sodium, so 2.8%, 0.2%. Calcium, so 3.6%, then 1.5%. Magnesium 2.1%, 0.1%. Then silicon 27.7%, then very negligible is present. So sulfur in non-living matter approximately 0.03%, in living matter 0.3%, sodium 2.8%, then 0.2%, then calcium 3.6%, in living matter 1.5%, then magnesium 2.1%, then 0.1%. Silica in earth crust highest amount is present 27.7%. In living matter are very negligible in a condition, but silica high amount is present in grasses, then grass leaf blades. These silica provide the stability of the leaves and some other uses also present. Then in cellular pool the average chemical composition is in cellular pool the average chemical composition water. So approximately 70 to 90% of water. In cellular pool, average chemical composition is water 70 to 90%. Next one, proteins. So 10 to 15% of proteins are present. Next one, nucleic acids. So 5 to 7% of nucleic acids. Then carbohydrates. So 3% is present. Next one, lipids 2%. Then ions are 1%. Are very traces amounts are present amounts. So average composition, chemical composition in a cellular pool, water 70 to 90%, proteins 10 to 15%, nucleic acids 5 to 7%, then carbohydrates 3%, lipids 2%, then ions 1% are present. So by performing the chemical analysis, we reveals the what type of chemicals are present in cell or cellular pool. So generally in a cellular pool, two types of chemicals are present. So these are inorganic chemicals and organic chemicals. In cellular pool, two types of chemicals are present. So these are inorganic chemicals, then organic chemicals. In cellular pool, two types of chemicals are present. So first one, inorganic chemicals, and second one, organic chemicals. So first one coming to inorganic chemicals. So inorganic chemicals generally, so water, then gases, then minerals. So inorganic chemicals generally water, gases and minerals. In plant body, minerals mainly present as a salts. In plant body, minerals generally present as a salts. Except carbon, hydrogen and oxygen, plants obtain these minerals from the soil by the absorption mode of nutrition. So that's why carbon, hydrogen, oxygen not considered as a true minerals. So except carbon, hydrogen, oxygen, plant obtain all these minerals from the soil. So in plant body, so generally minerals present in the form of salts. So inorganic chemicals do not possess carbon along with the hydrogen. That means carbon is not present in inorganic chemicals. These are not carbon containing compounds. So carbon not present in these inorganic chemicals. Next one, organic chemicals. These organic chemicals possess both the carbon and hydrogen. That means carbon present along with the hydrogen. So these are carbohydrates, proteins, fats, nucleic acids, vitamins, etc. etc. So organic are carbohydrates, then fats, then proteins, vitamins, then nucleic acids, etc. etc. These are the organic. So organic generally present in both the aqueous phase and non-aqueous phase. So organic chemicals present in both the aqueous phase and non-aqueous phase. all chemicals in cellular pool are obtained from the outer side. So all chemicals what are present in cellular pool, these are obtained from the outer side. So generally plant cell surrounded by the cell wall, then cell membrane, plasma membrane is present. So within this plasma membrane, cytoplasm and nucleus is present. So within cytoplasm different types of cell organelles are present. These are membrane bound and non-membrane bound cell organelles, except ribosomes in plant cell all are the single membrane bound or double membrane bound cell organelles. So these membranes, plasma membrane and cell organelle membrane maybe act as a semi-permeable membrane. So maybe act as a semi-permeable membrane due to this capability. Due to this capability or due to this nature, cellular pool have the capability to maintain the certain chemicals in each cell. Cellular pool have the capability to maintain certain chemicals within this cell in cellular pool due to nature of semi-permeable nature of plasma membrane and membranes of cell organelles. So due to semi-permeability nature, these will allow the certain chemicals from outside to inner side. So it is depending on metabolism of within this cellular pool or a cell organelle. So for example, chloroplast is mainly responsible for perform the photosynthesis. So what type of chemicals are required to perform the photosynthesis? So membrane of this cell organelle chloroplast and plasma membrane will allow these certain chemicals into cellular pool or within the cell organelles. So due to this semi-permeable nature, this will allow the certain chemicals from outside to inner side. So this semi-permeable nature will also useful to excretion of elimination of to permit chemicals from inner side to outer side are specific in a condition. So what are not necessary, etc, etc. This will allow to move from inner side to outer side. Due to this one, the cellular pool have the capability to maintain the certain chemicals within the cellular pool. It is entirely depending on metabolism, what type of chemical reactions are takes place. So these are cellular pool. So generally chemical analysis is a two types. So first one is a acid solubility test. Second one is a ash analysis. So chemical analysis is a two types. So first one acid solubility test, second one is a ash analysis. So first one coming to acid solubility test. So to perform acid solubility test, we have to take a living matter, any living matter, a piece of animal tissue or plant tissue, microbial paste or vegetable tissues. Then this piece of tissues are put or keep in a mortar and pestle. Mortar and pestle. Then by adding the chemical trichloroacetic acid. Trichloroacetic acid. Trichloroacetic acid. Grind this living matter. So this living matter is in mortar and pestle by adding chemical trichloroacetic acid. So grind this living matter. By grind we can get a thick slurry of this living matter. Then this thick slurry filter by grind this living matter we can get the thick slurry. Then filter filter this slurry by using a cheesecloth, muslin cloth or a cotton cloth. So by filtering this we can get two types of fractions. So first fraction is known as a acid soluble fraction. Acid soluble fraction or filtrate. Acid soluble fraction or filtrate. Second one is known as a acid insoluble fraction or regentate or pellets. So acid insoluble fraction, regentate or pellet. So first fraction is a acid soluble fraction. So filtrate. So in filtrate, what chemical compounds, both organic and inorganic, are dissolved in this water medium or chemical medium. These are present in acid soluble fraction. It is entirely cytoplasmic composition. Cytoplasmic composition. Acid soluble fraction is nothing but a cytoplasmic composition without the cell organelles are present. Then based on the solubility, molecular weight, size of the compounds. So compounds are two types. These are biomicromolecules. Biomicromolecules. Then biomacromolecules. Biomicromolecules and biomacromolecules. In acid soluble fraction, biomicromolecules are present. Biomicromolecules are present. In acid soluble fraction, biomicromolecules are present. These biomicromolecules are very simple, small. So these are small size of compound, simple chemical structure is present. Then molecular weight below 1000 Daltons are present. Below 1000 Daltons, approximately 80 to 800 Daltons are present. Approximately 80 to 800 Daltons. Then molecular conformation also, very simple molecular conformation is present. This is known as a biomicromolecules. Acid soluble fraction is nothing but cytoplasmic composition. In this biomicromolecules are present. So these are very simple chemical structure, smaller chemical compounds. Then molecular weight below 1000 Daltons, approximately 80 to 800 Daltons. Simple molecular conformation also present, chemical conformation. So examples: so water, then gases, then minerals, sugars, simple sugars, then amino acids, nucleotides, etc. So these are sugars, amino acids, nucleotides, then inorganic water, gases, minerals, etc. So biomicromolecules. Next, second one to acid insoluble fraction. In acid insoluble fraction, these are the large chemical compounds. So large chemical compound, then complex of structure, large chemical compounds, then complex of structure, then molecular conformation also very complex, very large in a condition, then molecular weight so greater than 1000 Daltons, approximately 10,000 Daltons or more than 10,000 Daltons are present. Molecular weight more than 10,000 Daltons. So these mainly cytoplasmic composition, along this one cellular organelles, cell organelles, acid soluble fraction, cytoplasmic composition majorly. Acid insoluble fraction, cytoplasmic composition, along this one cellular organelles, cell organelles are present. So these also nothing but biomacromolecules. Biomacromolecules. Example: carbohydrates, then fats, nothing but lipids, then proteins, then nucleic acids. So carbohydrates, lipids, proteins and nucleic acids are present in acid insoluble fraction. Acid soluble fraction may contains both organic and inorganic. Then acid insoluble fraction mainly contain organic compounds. In acid soluble fraction, some inorganics, sulfates and phosphates. So sulfates and phosphates are inorganic elements. So these are maybe present in acid soluble fraction along with the organic compounds. So by performing the elemental analysis, we can get the list of elements. So what are present in a cellular pool. Then by performing the compound analysis, so we can get the list of organic and inorganic compounds. So what are present in living tissues. Next one, ash analysis. For performing the ash analysis, here also we have to take a living tissue, any living tissue. Then completely dry this living tissue in shade condition, shade dry. So in fresh condition, it have the weight. So along this water, that means moisture content. By shade drying, evaporates the entire moisture content, water content. This weight is known as a dry weight. This weight is known as a dry weight. Generally, 4 elements. So carbon, hydrogen, oxygen and nitrogen. So 99% in substances these 4 elements are present: carbon, hydrogen, oxygen and nitrogen. So in 99% of substances 4 elements are present: carbon, hydrogen, oxygen and nitrogen. These are 4 types of elements combined in a different formation and different ways from the biomolecules. So biomolecules are building block for life and perform the all physiological activities. So 95% of dry weight. 95% of dry weight composed or contained these 4 elements: carbon, hydrogen, oxygen and nitrogen. In this more than 50% is a carbon. More than 50% is a carbon is present. Then to perform the ash analysis, we have to take the living tissue, then dry this entire living tissue. By drying, we can get the dry weight. That means evaporates the moisture, water content from this fresh material, living material. By this drying, we can get the dry weight. Then burn. So burn this entire dry material. By burning, so burn oxidize the all organic compounds. By this burn oxidize all organic compounds. That means carbon compounds. The living material is known as a ash. The living material is known as a ash. So this ash contains inorganic elements. This ash contains inorganic elements. Ash contains inorganic elements. So by completely burnt, oxidize the all carbon containing compounds. the living powder like a material is known as ash. Ash is nothing but of inorganic elements. So by performing ash analysis we can get the list of elements so what are present in a living tissue. If we perform this elemental analysis by using the earth crust also, we can get the same list of elements what are present in living matter and non-living matter, but the relative abundance may be vary from one element to another element in living matter and non-living matter. So by performing elemental analysis we can get the list of elements. By performing the compound analysis we can get the list of the compounds both organic inorganic compounds what are present in a cell. Then by performing the analytical techniques, some tools and techniques, we can know the molecular formula, then compound structure. We can separate compounds from one compound to another compound also. So what chemicals biomolecules are participate in metabolism in chemical reactions. By participation in metabolic reactions these biomolecules what are participating in metabolic reactions constantly change from one form of biomolecules to another form of biomolecules. These biomolecules is nothing but metabolites. These biomolecules is known as a metabolites. So metabolites is a nothing but biomolecules. Metabolites is nothing but biomolecule. These are formation by the chemical reactions of biomolecules. So biomolecules participate in chemical reaction. So constantly change from one form to another form. So micromolecules and macromolecules. So generally lipids. So lipids molecular weight less than 1000 Daltons are present. So lipids molecular weight is less than 1000 Daltons, but these lipids are present in biomacromolecules. Biomacromolecules. Why? Because when grade these living matter. So generally lipids are water insoluble condition. So these get fractionate or differentiate or break into small fragments like a condition. So these fragments of lipids present along with the fragments of cell membrane. So non-polar ends of lipids combines with the fragments of plasma membrane. So lipids generally present in biomacromolecules, but molecular weight is less than 1000 Daltons are present. So why? Because when we grade this living matter, lipids generally present in plasma membrane. So non-polar ends of this plasma membrane combines with fragments of plasma membrane. So generally these lipids are not soluble in water. So due to this reason these are present in acid soluble fraction. But lipids are not strictly biomacromolecules. Lipids are not strictly biomacromolecules. Generally biomacromolecules function as a structural components. Structural components. So biomacromolecules function as a structural component, then nutrient stores. Nutrient stores, then storehouse of genetic information. Storehouse of genetic information, then energy sources, then molecular messengers, then enzymes. So biomacromolecules act as a structural components, nutrient stores, storehouse of genetic information, energy sources, molecular messengers, and enzymes. Then in biomacromolecules, except lipids, except lipids. All are formation by the polymerization. All are formation by polymerization or condensation. Polymerization or condensation of subunits. These are called monomers. These are called the monomers. So by the polymerization of these monomers, by combining these monomers, form these large macromolecules. These are nothing but polymers. These are nothing but polymers. That means carbohydrates, proteins, nucleic acids. These all are polymers, but lipids are not polymers. Why? Because except lipids, all macromolecules formation by polymerization of monomers. That means subunits. By condensation of these monomers form a different length of polymers. Different length of polymers, different length of chains of polymers. It is depending on number of monomers are present in this polymers. So metabolics, these are macromolecules and micromolecules are nothing but metabolites. So metabolites are basically two types of primary metabolites and secondary metabolites. So primary metabolites and secondary metabolites. So first one coming to primary metabolites. So by these biomolecules participate in metabolic reaction. So first formed metabolites is known as a primary metabolites. These primary metabolites are essential for life of organism. Without these primary metabolites, organisms cannot complete its life cycles. These are known as a primary metabolites. Examples: carbohydrates, then proteins, then lipids, chlorophylls, then heme, group of carbohydrates, proteins, lipids, chlorophylls, heme, then nucleic acids, etc. Primary metabolites. So without these macromolecules, chemicals, metabolites, organism cannot complete its life cycle. These are necessary to complete its life cycle. These primary metabolites are present in all types of the organism, plants, animals, microbes, etc. So for example, nucleic acids are necessary for completes its life cycle for all organisms. Genetic material, without genetic material, organisms cannot evolve, cannot continue its life cycle from one generation to another generation. At the same time, cannot express the physical characters, genetic characters. So these are present in all types of the organisms, animals, plants, microbes, all types of the animals. So these primary metabolites have the known function. Primary metabolites have the known function, then identifiable physiological function in a living organism, life cycle of organism. So primary metabolites has a known function, identifiable physiological function in a life cycle of organism, like a photosynthesis, respiration, protein metabolism, lipid metabolism, etc. etc. Then these primary metabolites are present in throughout the life cycle of organism. So for example, plants, from germination to then death of the plant, natural germination to natural death of the plant. So these are present in throughout the life cycle of organism. At the same time, these primary metabolites are present in all types of the plants. All types of the plants, there is no exemption. So that means in entire plant kingdom, in entire plant kingdom. So these primary metabolites are present in entire plant kingdom. That means in all types of the plants. Next one, primary metabolites plants produce the larger quantities. That's why extraction also very easy in a condition. Extraction also very easy. So plant body or animal body produce the large quantity of these primary metabolites. That's why extraction also very easy in a condition. Then these metabolites are stored large quantities in all parts of the plant bodies. Next one, secondary metabolites. Secondary metabolites. Generally, some plants, then microbes and fungi. So plants, fungi and microbes. So plants, fungi and microbes produce the number of secondary metabolites. So these secondary metabolites are derivatives of the primary metabolites. So primary metabolites are participate in a metabolic reaction. So on that time maybe produce some other form of biochemicals. These are known as secondary metabolites. These secondary metabolites mainly appears in plants, fungi and microbes, very rare in animals. So secondary metabolites are derivatives of primary metabolites. So these are resins, gums, then antibiotics, some pigments, drugs, toxins, then polymeric substances, etc. Where these secondary metabolites are produced small quantities. Secondary metabolites are producing in small quantities. That's why extraction also very difficult in a condition. Then secondary metabolites stores in plants smaller quantities, are not stored in all plant pods. Secondary metabolites are stored in small quantities, so that's why extraction is very difficult in a condition. At the same time, secondary metabolites are stored in all plant pods, all parts of the body of organism. At the same time, these secondary metabolites not have the identifiable function, known physiological function in a life cycle of an organism. Primary metabolites directly participate in a metabolic reaction or physiological function of organism, but these secondary metabolites not directly participate in a metabolism of an organism. These are not essential to complete its life cycle of an organism. At the same time, these secondary metabolites not present in all types of the plants. That means one type of secondary metabolites not mandatory, not present in all types of the plants. At the same time, all parts of the plants also. So primary metabolites are present in throughout the plant kingdom, in all types of the plants from birth to death. But secondary metabolites are not present in all types of the plants and all parts of the plants. These are not present throughout the plant kingdom. That means from natural birth to natural death. These may be derivatives in middle of the life cycle of organism or end of the life cycle of organism. Maybe at any time. It's depending on life cycle, habitat of the plant bodies. And also these not have any known identifiable functions. Secondary metabolites. So some secondary metabolites are pigments. So generally chlorophylls are pigments, but chlorophylls are necessary to perform the photosynthesis. So those are primary metabolites. So pigments are other than chlorophylls. These are carotenoids, then carotenoids and anthocyanin. So generally anthocyanin is present in vacuole of the plant cell. Anthocyanin is present in vacuole of the plant cell. These are red color, blue color, then purple color. It is responsible for colorization of different plant pods. Next one, alkaloids. Alkaloids are morphine, then codeine, etc. Next one, terpenoids. So these are monoterpene, then diterpene. So terpenoids are monoterpene and diterpene. Then essential oil, lemongrass essential oil, groundnut oil, castor oil, etc. Next one, drugs, heparin and resin. Drugs are heparin and resin. Then lectins, concanavalin A. Lectins are concanavalin A. Then toxins, then drugs, vinblastine, then curcumin. So drugs are vinblastine and curcumin. Then polymeric substances. So polymeric substances. So rubber, then gum, then cellulose. So some examples are secondary metabolites, pigments, carotenoids and anthocyanin, alkaloids, morphine, codeine, terpenoids, monoterpenes, diterpenes, essential oil, lemongrass oil, toxins, heparin, resin, lectins, concanavalin A, drugs, vinblastine, curcumin, then polymeric substances, rubber, gum and cellulose. So these secondary metabolites are isoprene compounds, then nitrogen compounds, then phenolic compounds, etc. Then what are the uses of these secondary metabolites? So uses of secondary metabolites. So secondary metabolites generally be useful to attract the pollinators. These are useful for pollination purpose. So secondary metabolites will useful to attract the pollinators, will useful for pollination. At the same time, these are will useful for seed dispersal. These are will useful for the seed dispersal system. Then secondary metabolites will useful to provide the protection from herbivorous animals. Herbivores provide the protection from herbivores. Then pathogens, herbivores and pathogens. Then secondary metabolites useful to produce the number of industrial compounds. So these are drugs, then insecticides, antibiotics, drugs, insecticides, antibiotic, then scents, then spices, gums, etc. etc. So secondary metabolites will useful to produce the number of industrial compounds. So generally in chemistry point of view, these are compounds are classified as aldehydes, phenols, ketones, etc. So aldehyde, ketones, phenols, etc. etc. In biological point of view, these are known as classified as carbohydrates, proteins, lipids, nucleic acids, etc.